Hi everyone, in today's class, we will start the chapter Cell Cycle and Cell Division. Look at these two babies, cute na? See, to study cell division, we first need to know why cell division occurs. A cell is the basic unit of life. हम सभी बचपन से पढ़ते आए हैं कि सेल हमारे लाइफ की बेसिक स्ट्रक्चरल और फंक्शनल यूनिट है. A small cell divides countless times to form a complete organism. And you know that in case of prokaryotic cell, a single cell is a complete organism. So, cell division is important for the growth and survival of every organism. And cell division is equally important to the survival of our species on Earth. Now, we know that our cells contain our genetic material DNA and all informations is present in our DNA for everything that our body needs to function in order to survive and what our external structure will look like. So, it took millions of years to evolve all this information contained in our cells. That's why all this information is transferred from the parent cell to the daughter cells throughout cell division. Okay, तो आप समझ गए होंगे. Now we will discuss the process of cell division. First we will see how cell division occurs in prokaryotic cells and then we'll see how eukaryotic cells divide. If you don't know difference between prokaryotic and eukaryotic cells, then watch this video of mine. I will provide the link. Think of it as a bacterial cell. We all know that bacterial cells or prokaryotic cells do not have any membrane bounded organelles. So, they don't have a nucleus and genetic material of bacterial cell यानी कि DNA is contained within its cytoplasm. Hence, their DNA is called nucleoid. So now, see how cell divide in their case. Prokaryotic cells do not undergo mitosis. और उनके केस में किसी भी प्रकार का सेल साइकिल नहीं होता है. In their case, direct cell division occurs. So, their process of cell division is called amitosis. First of all, DNA replicates within its cytoplasm. It means that the amount of DNA becomes double, and as the DNA replicates, the cell divides from the middle. That means there is no karyokinesis or cytokinesis in their case. And since DNA replicates just before fission, hence DNA replication in bacteria is known as prior to fission. This question can come as MCQ, that how DNA replication happens in bacteria. Then answer will be prior to fission. That means cell division process of prokaryotic cells is very simple. Here only DNA is replicating, then the cell is dividing into two daughter cells. So what do you understand here? Growth and reproduction are the same thing in prokaryotes. क्यों? क्योंकि growth cell division से होता है और reproduction भी cell division से होता है. And prokaryotic cell means unicellular cell where a single cell is an organism. That means जब organism की growth के लिए सेल को विभाजित किया जा रहा है तो उस सेल डिवीजन के माध्यम से एक साथ दो नई सेल का निर्माण भी हो रहा है. इसका मतलब है कि ग्रोथ और रिप्रोडक्शन दोनों प्रक्रिया एक साथ साइमल्टेनियसली हो रही है. ओके, नाउ लेट्स सी यूकैरियोटिक सेल डिवीजन. This process is comparatively a little bit more complicated, but very easy, don't worry. A eukaryotic cell can divide in two ways, mitosis cell division and meiosis cell division. ठीक है, चलो देखो. एक सेल डिवाइड होने के लिए दो फेज से गुजरती है, इंटरफेज एंड माइटोसिस फेज और एम फेज. Interphase is the phase where a cell prepares itself to divide, and M phase is the phase where a cell finally divides to form new daughter cells. Interphase typically takes much longer time compared to M phase. ऐसा माना जाता है कि 95% of the time is spent in interphase state. How long it takes for a cell to divide depends on each organism and cell type to cell type. जैसे कि ह्यूमन सेल को आर्टिफिशियल मीडियम में डिवाइड होने में 24 आवर समय लगते हैं. Your NCERT says cells in culture. It means artificial medium. अगर आप जूलॉजी में BSc करते हो तो आपको टिश्यू कल्चर, सेल कल्चर पर प्रैक्टिकल करना होगा. एंड यू नो इट्स वेरी इंटरेस्टिंग. तो आई वॉज सेइंग ह्यूमन सेल को आर्टिफिशियल मीडियम में डिवाइड होने में, यानी कि इंटरफेज प्लस एम फेज, टोटल सेल साइकिल कंप्लीट होने में 24 आवर समय लगते हैं. और ईस्ट, जो कि एक यूकैरियोटिक सिंगल सेल्ड माइक्रो ऑर्गेनिज्म है, ईस्ट के सेल साइकिल कंप्लीट होने में 90 मिनट समय लगता है. So, duration of cell cycle can vary from organism to organism. And not only that, different type of cells present in an organism's body may require different time duration for divide. Hope it's understood. Now, let's discuss the interphase. Cell cycle is a little bit complicated process. So, for easiest understanding, we have divided the cell cycle into different subphases, as interphase is divided into 3 more subphases. These are G1 phase or gap 1, S phase or synthesis phase, G2 phase or gap 2. Now, let's see how a cell prepares itself to divide. During G1 phase, a cell grows. This means that the cytoplasm increases. And if the cytoplasm increases, then what will happen? Compared to before, the nuclear cytoplasmic ratio will decrease. इसके अलावा कुछ ऑर्गेनल्स साइटोप्लाज्म के साथ बढ़ते हैं, जैसे गोल्गी बॉडी, एंडोप्लाज्मिक रेटिक्यूलम. ये ऑर्गेनल्स दुगने हो जाते हैं. And when the cell is in the G1 phase, damaged DNA in the nucleus is also repaired. It is a very important function that occurs in G1 phase because S phase comes after G1 phase where DNA replication takes place. इसलिए यदि डैमेज DNA S phase में जाता है तो डैमेज DNA replication बन जाएगा और डॉटर सेल में abnormalities देखी जाएगी. That's why damaged DNA repair is a very important function of the G1 phase. इसके अलावा G1 phase में transcription and translation भी होता है, क्योंकि each phase of the cell cycle requires enzyme to carry out all metabolic functions in our body. And how enzyme forms through transcription and translation. तो समझ आया कि G1 phase में क्या changes हो रहा है. अब हम देखेंगे कि इस फेज में क्या चेंज होते हैं. चलो, अब इस फेज यानी कि सिंथेसिस फेज. क्या तुम नाम से समझ सकते हो कि यहाँ कुछ सिंथेसिस होगा, यानी कुछ बनाया जाएगा? यस, इस फेज में डीएनए सिंथेसिस होगा. दैट इज, डीएनए विल रेप्लिकेट एंड क्रिएट अनदर डीएनए स्ट्रैंड लाइक इटसेल्फ. व्हाई डज डीएनए रेप्लिकेशन टेक्स प्लेस? Parent cells divides to form 2 daughter cells, and to pass the genetic material, that is DNA, equally in both the daughter cells, the DNA is duplicated by DNA replication. Okay? And this DNA replication occurs inside the nucleus. और DNA डबल होने के साथ हिस्टोन प्रोटीन भी डबल हो जाएगा. Because we all know that histone proteins provide structural support to DNA. Then the newly formed DNA also needs supports, so the histone protein also doubles. And another function that occurs only in animal cells, that is centriole, is a membraneless organelle found in animal cells. This centriole also doubles in S phase. Okay, hope it's understood. Our next phase is the G2 phase or gap 2 phase. Since this phase is the next phase of the synthesis phase or S phase, so it is called the post-synthetic phase. And since this phase is the previous phase of mitosis phase, it is also called pre-mitotic phase. At this phase, the cell prepares itself to enter the M phase. M phase में spindle fiber बनते हैं. And the protein required for the formation of this spindle fiber is produced in G2 phase. This protein is tubulin protein. इसके अलावा ट्रांसक्रिप्शन एंड ट्रांसलेशन होता है क्योंकि एम फेज में हमें ज़्यादा एनर्जी चाहिए. Besides, organelles like mitochondria, chloroplast, plastid, etc. duplicate in G2 phase. Okay, so interphase states end here. Sorry students, today's video is getting too long, so I will cover the mitosis phase in the next video because my phone doesn't have enough storage for such a long video. I'm so sorry. Don't worry, I will teach you the mitosis phase very soon. I will add it to the playlist. Please check it out. Bye for today. See you in the next video. Thank you for watching.